Hello everyone, so this is a walkthrough of Combined Science Trilogy, Biology Paper 1 Foundation tier, and this was the paper that was sat in 2022. Okay, so I'm going to run through this, go through how I would have tackled it, and then I've put little revision videos in the description for each different question if you need to go back and revise that content. I'm going to try and go through it relatively efficiently, but yeah, hopefully you can use this to identify what areas that you need to focus on. So question number 1 then. Foods are digested before they are absorbed into the blood. Figure 1 shows the organs in the human digestive system. You've got these different labels here. So before I even start off, I would label it, okay, just to see if I can recall this knowledge before I look at the question. So A would be the mouth, B, the biggest organ here is the liver, C would be the stomach, and then D is pointing to the small tube, which would be the small intestine. Okay, so looking at question number 1, which organ is the stomach? We've already done that, it would be C. What is the type of enzyme produced in the stomach? Well, this is some very precise knowledge that you've got to know quite early on. You've got to know where these different enzymes are produced. So carbohydrates are produced in your saliva, the pancreas, and small intestine. Lipases are only produced in the pancreas and small intestine. But proteases are produced in the stomach, the pancreas and small intestine. So therefore, the answer would be protease. All the types of enzymes are always produced in the pancreas and small intestine, but the difference is proteases are also made in the stomach, carbohydrates are also made in the saliva. Question 1.3, which term describes the pH in the stomach? Give one reason why the stomach is this pH. Well, it is acidic, you should know, because the stomach has acid inside it. That type of acid is called hydrochloric acid. Okay, and therefore it is the optimum pH, meaning the best pH for protease. Which organ produces bile? Well, bile is a substance that you've got to know is made by the liver, and it is stored in the gallbladder. So remember, liver makes it, but also be aware it's stored in the gallbladder. How does bile help the digestion of foods? Well, you've got 3 options here. It's either increasing the surface area of fats, it's an enzyme that digests protein, and it makes the pH in the small intestine more acidic. Okay, well, first of all, bile itself is not an enzyme. It's just a digestive juice that helps brings digestion about, so it's not an enzyme. It is actually also an alkaline substance, so therefore it's not going to make it more acidic. So what bile actually does is break down the fats, increasing the surface area. This process is called emulsification. Imagine if this is my fat blob that I'm needing to digest, the bile comes along and breaks it down into smaller pieces that then the lipase enzyme can come along at and then help chop it up into smaller pieces. And this whole process of the breaking down of fat, emulsification. A student tested different foods for the presence of protein, starch and sugar. Draw one line from each food molecule to the reagent used for that food molecule. So this is just knowing your food tests. The first one is protein, and you just need to know we use burette reagent to test for protein. To test for starch, we use iodine solution. And finally, we use sugar. As to test for sugar, we use Benedict's solution. So those are the different solutions that you need to know. There is also one more solution that isn't listed. Lipids, we use ethanol, which is a clear, colourless liquid. Give one safety precaution a student could use when using Benedict's solution. Well, any time, doesn't have to be Benedict's, but if you're using a solution, the easiest one marker for safety is just saying wear goggles. Because any solutions going into the eye is obviously a bad thing. Question 1.8, Table 1 shows the results for one food sample. Which of the tests show positive results? So this is when you've got to know your colour changes. So for Benedict's starts off blue, and if sugar is present, it will either turn orange or red. So this would be a positive result. Burette's solution also starts off blue, but because it hasn't changed and it's still blue, that means it would be a negative result. Iodine is an orangey colour. If you add iodine to starch, it should go a blue-black colour, and because it has changed black, yes, that would be a positive result. So it would be Benedict's and iodine only that then give us positive results. Starch molecules are not absorbed into the blood from the digestive system. Give one reason why. Well, going back to the whole point of the digestive system is breaking down large, complex molecules into smaller ones. So this could be modelled as a representation of starch, and what we do is break it down into glucose to absorb it into our body. So you would just simply be starch is too big to be absorbed through the walls of the small intestine. We have to break down starch, we have to break down protein, and we have to break down lipids for those substances then to be absorbed into our body. Okay? Right, question number 2 then, moving on from digestion, is looking at plants, and it's showing us a cross-section through a leaf. So this topic is knowing the structure of the leaf and its adaptation for it. So you've got, give one way that the palisade layer is adapted for photosynthesis. So you should know that in a leaf, most of the photosynthesis happens in the palisade layer. You can see here it's got loads of these grey dots. You should be thinking, what are these little grey dots called? Well, you know, photosynthesis. What does a plant need for photosynthesis? Yes, it needs light, CO2 and water, but to absorb the light, it needs the chloroplasts. And the palisade layer has loads of chloroplasts to absorb the light. That's the function of the chloroplasts, the chlorophyll inside it, absorbing all of the light for the energy for photosynthesis. The gases pass into and out of the leaf through small pores on the surface of the leaf. What are the small pores labelled X called? We should know that the little holes on the underneath, the actual hole is called the stomata. Okay, and the stomata are opened and closed by the guard cells. So normally, kind of represented like this. You have the guard cells, okay, on the bottom of the leaf, and they can open or close the actual physical hole, which is the stomata. So in the day, guard cells are normally open, larger, and that allows the stomata to be open. At night, the guard cells shut, and therefore there's no gap, no stomata are open, and therefore no water is lost. A student viewed a section of a leaf using a microscope. The student measured the length of one of the palisade cells. The cell image measured 28 millimeters in length when viewed at a magnification of times 400. Calculate the real length of a palisade cell in millimeters. Okay, so you are after, and it's given you the equation, the real length is image divided by magnification, and it's always in millimeters. So this should be a relatively simple one. It's just substituting in what you've been given. So the image length, you can see here, is 28 millimeters. Your magnification from the question is 400. Now normally I'd be looking for a unit conversion here, but as there isn't one, I would just simply type in on my calculator 28 divided by 400, and that gives me 0.07 millimeters. Now there's no other tricks on this question, so I just type that in and that is my answer there. It then says, convert the real length of the cell from millimeters to micrometers. Well, in that case, to convert it, what you have to then do is remember that it's even told you here, so you don't need to remember. In fact, it's told you 1 millimeter is equal to 1,000 micrometers. So to convert it, you are timesing by 1,000. So you take your answer of 0.07 and you times it by 1,000, and that would give you an answer of 70 micrometers. So 0.07 millimeters is the same as 70 micrometers. Carbon dioxide can move into and out of cells. What is the process by which carbon dioxide can move in and out of cells? Well, it's carbon dioxide, so it's not osmosis because that's only water. Active transport is when it goes from low to high concentration, so it's not that. So it would just be the simple method of diffusion. Diffusion is the net movement of substances going from high to low concentration. Figure 3 shows a diagram of 4 cells, A, B, C and D, and you've got the carbon dioxide molecules are represented by the dots. Okay, and it says, which cell will carbon dioxide move into at the fastest rate? And give a reason for your answer. So we think back to our definition of diffusion. Diffusion, movement of particles from high to low concentration. And we want to know which way is it going to go in to the cell. So it's saying which one has the biggest difference between the concentration outside and inside. Well, if I look at my first one, cell A, I can see that there is a clear difference. The particles will move from high to low concentration going in. If I look at B, there's a kind of even concentration here, so the particles wouldn't necessarily move in. Same with C, there's an even concentration. And on D, the particles are actually higher inside than outside, so the particles will move out of the cell. But we've been asked about what one would go in, so therefore it would be cell A. And you give a reason for your answer, because there's a higher concentration of particles outside than inside, therefore particles move in. Okay, and to be honest, you don't need to even say it to that detail. You just say there's a bigger difference in concentration outside than inside. A student then investigated the effect of different colours on the rate of photosynthesis. Figure 4 shows some of the apparatus used. Okay, now you should be thinking, oh, remember this practical with pondweed and being and counting bubbles underwater. However, you probably at school would have changed your light intensity by moving your lamp. They are not changing light intensity, they are changing the colours. So I would be thinking straight away, right, my variables, what am I changing? My independent variable is my colour of light, because that is what they are changing. They are going to be measuring either the bubbles of gas, or they're going to be measuring the volume of gas made. And then you should be thinking about, right, what are they going to keep the same? What are their control variables? Well, if they're changing the colour of light, you would need the light intensity to be the same. you'd have the same amount of CO2 potentially, but also the same temperature of the solution, of the water, because temperature affects photosynthesis as well. The student placed this apparatus in blue light, then green light, and then red light. The student measured the rate of photosynthesis in each colour of light. What two measurements should the student make to calculate the rate of photosynthesis? Now, in this one, you can actually say, well, you need to know about the actual bubbles. So it's effectively, what do they need to measure? Well, they need to measure how many bubbles are made, or the volume of gas that is produced. So you could say amount of bubbles. A slightly more scientific way would say the volume of gas produced, of oxygen. But the next one is about the rate. Now, if you want to calculate, the rate is basically saying how fast it is. If you want to know how fast it is, you've also got to know the time as well. So it would be amount of gas divided by time, or if you went to bubbles, divided by time to give you the rate. Give two control variables. The student should have kept the same in the investigation. Where if you had done a little brainstorm here at the top, you've already got the answers. They're changing the colour, so you would need the light intensity to be the same and the temperature to be the same. So whenever you get these sort of questions, do a little brainstorm before you even think of, read the questions, and odds are you'll have the answers already. So light intensity and temperature. Table 2 shows the results. So you've got the colours of light and you've got the rate of photosynthesis in arbitrary units. Arbitrary units is just what they say when you don't need to know what actual the units are. Complete figure 5. We need to label the y-axis, use a suitable scale, plot the data and label each bar. So as you do this, tick off the bullet points on a graph question to make sure that you've done them all. Okay, because people might go straight to plotting the data and miss the first one, label the y-axis. So you look at your table they've given you, you know colour's going to be at the bottom, so rate of photosynthesis in arbitrary units would be going on your y-axis, and copy it exactly the same. So rate of photosynthesis in arbitrary units. Okay, copy that whole box, and you know you've got colours at the bottom. Now, because we've got colours here, we've got words, it means we've got categoric data. Okay, and because we've got categoric data, that means we would draw a bar chart. So we've got blue is 9, green is 1, and red is 8. Okay, so we then need to come up with a scale, and we know we've got to get up to 9. So I would just keep it simple, and also they tend to give you the right number of squares to try to help keep it simple. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, could go up to 10 as well. Okay, so they've given you a quite nice obvious scale, and we need to know that blue goes up to 9. So blue, you're going to draw a line. Obviously you would be using a ruler, making sure this is really accurate. Go exactly to 9, draw your lines on. Okay, then you could do green, I believe. Green is just 1. Go up to 1, exactly, draw your lines on. And then red, just double check it, is 8. So again, you would just go up to 8, draw your bar on, and because it's a bar chart, we leave a little gap between each one, and that's 4 easy marks there. We have labelled the y-axis, used a suitable scale, plotted the data, and then labelled each bar at the bottom. Okay, so just make sure you've done that as well. That's 4 marks just through drawing a very simple bar chart. Look at table 2. What colour of light should be used to grow plants in a greenhouse? Well, we want to have the highest rate of photosynthesis. The highest one is our blue light. So we should choose blue light because it's got the highest rate. Question number 3 then. Different topic. This question is about disease. Rose black spot is a disease where black spots develop on the leaves of rose plants. What type of pathogen causes rose black spot disease? Well, rose black spot is one of the diseases. There are 7 diseases you've got to know, and this is your one fungal example. Your bacterial diseases that you've got to know are salmonella and gonorrhea. Rose black spot is the fungal one. Malaria is your protist disease. And then you've got to know 3 viruses, which are measles, HIV, and for plants, tobacco mosaic virus. Question 3.2. Plants with rose black spot disease often have yellow leaves. Suggest one reason why the leaves are yellow instead of green. Well, you'd be thinking, well, what part of a plant, or what actually makes a leaf green? It would be the chlorophyll. The chlorophyll is what makes a leaf green. So maybe if it's yellow, it doesn't have enough chlorophyll. It's got less chlorophyll. So it's less chlorophyll. Okay, or you could say less chloroplasts. Question 3.3. Explain why plants with yellow leaves will grow more slowly. Well, if they've got less chloroplasts, as we've just said, that means that less photosynthesis will be occurring. So less photosynthesis occurs due to less light being absorbed. Okay, so that would be that is your 2 points there. Less photosynthesis is occurring because less light is being absorbed, but you could also add in that therefore, because there's less photosynthesis, less glucose would also be made. Because remember, photosynthesis is making glucose for the plant to respire and use it for growth as well. Question 3.4. The spread of rose black spot can be controlled by using different methods. Draw one line for each method of control to how it works. So, removing and burning infected leaves. How does that actually stop rose black spot from spreading? Well, would it create a barrier? Well, if you're burning them, that's not creating a barrier. But if you're burning them, are the pathogens getting killed? Yes. So this one, removing them and burning them, yep, absolutely. And then it says, water the roots of the plant, not only the leaves. So if I'm watering the roots, okay, is that making a barrier? No. If it reduces the chance of the pathogens being spread by water droplets, well, that kind of makes sense. If I'm just watering the roots, I'm not watering the actual leaves. That means there's be less chance of water spreading it. So I feel quite confident it would be that one. But you could check the bottom one, reduces the temperature so that pathogens reproduce less, or just by watering it wouldn't affect the temperature, so it's not going to be that one. Question 3.5. Tobacco plants may be infected by the other pathogen I mentioned called TMV. What type of pathogen is TMV? Is it all in the name? It's tobacco mosaic virus, so it's just simply virus. Malaria is a disease caused by a protist. How is the malarial pathogen transferred to humans? Okay, so you've got to think about what do you know about malaria. How does malaria effectively is spread? Well, it's by mosquito or mosquitoes biting humans. And therefore the protist is therefore exchanged between person and person. And because we know that, how can the spread of malaria pathogens be reduced? So we've got to know, right, we know it's spread by mosquitoes, so which of these would stop the mosquitoes? Avoiding sexual contact. Well, that wouldn't stop mosquitoes. Cooking food thoroughly? No, that wouldn't stop mosquitoes. Draining water from swamps? Maybe. Use a tissue when sneezing. That wouldn't stop mosquitoes, so therefore a more logical one would be this one, because if you destroy the swamps, mosquitoes live and breed in stagnant water. If you destroy the swamps, you destroy their habitat. You destroy where they live, and therefore they can't reproduce. So that is how it would reduce the spread of malaria. Question number 4 then. Cigarette smoking is one of the main causes of cancer in the UK. Mutations in cells cause cancer. Where in a cell do mutations happen? Okay, so you've got the cell membrane, the cytoplasm, or the nucleus. And you should remember, mutations is a change in DNA. Okay, that is what a mutation is, and you've got to think, where is your DNA held in a cell? Well, it would be the nucleus. Remember, all your DNA is contained in the nucleus. Why do some cancers develop into large tumours? Okay, so you've got to think, right, what do I know about a tumour? Is it because cells never stop dividing, cell respiration is slowed down, or enzyme activity is stopped? Well, if you've got respiration slowing down, or enzyme activity being stopped, that would stop cell division and growth. Okay, so it's not going to be those two. A tumour is caused by uncontrolled cell division, cells that do not stop dividing. Cigarette smoking has been linked to many types of cancer. Lung cancer is the most common type of cancer caused by smoking. Suggest one reason why. Well, it's just a 1 mark suggest question. Try and use a bit of logic with this. When you are smoking, where is all that chemicals going? Chemicals are going into your lungs. Okay, so chemicals are inhaled into the lungs, and that's why it affects your lungs and causes lung cancer. Or chemicals enter the lungs first. Question 4.4. A person with lung cancer can develop secondary cancers in other parts of the body. Describe how this can happen. Okay, so what you need to know in this part is basically how you get your primary cancer, your tumour, okay, and then what then happens to cause cancer to spread. Well, what happens is, how does the tumour pieces move about the body? It happens through the blood. So you could say a part of the tumour breaks off and travels in the bloodstream, where it then be developed or deposited in a different part of the body, and it can continue dividing into a different tumour. Sometimes a person may need a lung transplant. The National Health Service will not offer a lung transplant to a person who smokes. Give a reason why. Okay, well, this again, suggest a reason why. So you've got to come up with a logical reason. So, where if they're smoking still, it will just damage, smoking will damage the new lung. You could also say other things about cost, about there just being limited finances. You could then also say it could be used for someone else who is not smoking. Okay, and therefore not damage it. Figure 6 shows data about skin cancer in males for different age groups in the UK. The data shows the number of new cases of skin cancer in 1 year. Okay, so you've got the number of cases of skin cancer, and you've got the different ages in years. How many more new cases of skin cancer are in males 40 to 44 than 15 to 19? Well, to use this, you've got to look at your graph. So 15 to 19, and then you've got to see, right, this is my first bar here, and it's gone up one little square. You know, if you're going from 0 to 100, and there are 5 little squares. So 100 over 5 means that each little square is worth 20. So therefore in 15 to 19, it's just 20 new cases of skin cancer. In 40 to 44 though, you see we track the line along. It's one little square up above 300. So this one would be 320. And it's saying how many more cases. So it would be 320 minus 20. That's the difference, which would give us an answer of 300. Question 4.7. There are no new cases of skin cancer diagnosed in males younger than 15. Suggest one reason why. Well, if you're younger than 15, odds are you're still quite young. You haven't been exposed to enough radiation to then cause it. Okay, so you could say again it could be any logical reason why, but you just say little exposure to radiation, which would then cause mutation, or limited exposure to sunlight. You could even say using more sunscreen, which would then block out those harmful UV rays. Okay, and then give one conclusion from the data in figure 6. So you go back to figure 6 and look at the conclusion, where you can see what is the general trend. As age increases, there are more new cases of skin cancer up till about 65 to 69, and then the number of cases then start to go down. Okay, now you could just say, because it's only a 1 mark question, you can just say about the biggest bar. Okay, the most common age to get new cases of skin cancer is between 65 to 69, or you could break it down into what I've just said. It increases each year up to 69 and then decreases from 69 to 90. So I would just say the most common age of new cases is 65 to 69. You could say the numbers increase between 15 to 69 and then decrease between 69 and 90. So although this was only a 1 marker, if it was a 3 marker, you've got loads of different things that you could have also said as well. Survival rates for all types of cancers have improved over the past 20 years. Give 2 reasons why. Well, why are we getting better at treating cancer? Well, we've got improved treatment and drugs. And medicines to help do that. And the other thing that we're doing, we're screening a lot earlier and are much more effective at detecting it earlier. So we've got earlier diagnosis, or you could put identified earlier. Okay, those would be your 2 key points on that. Okay, so question number 5 then. Bacteria can cause a variety of diseases in humans. What are 2 similarities between a bacterial cell and an animal cell? So the first thing I would do is just draw out, right, this is going to be my main structure of my animal cell, and then my bacterial cell. Okay, so we've got just the DNA free, we've got some little flagellum coming off the end, and then we've got, remembering they've got 2 layers on the outside, and then I can just quickly label them, and I know I've got my nucleus, my cytoplasm, my mitochondria. The smallest ones are the ribosomes, and going all around the outside is the cell membrane. Then we've got a bacterial cell. It has a cell wall. The inner one is the cell membrane. Some bacteria have flagellum, not all, but some do. And then you've got the free DNA, not in a nucleus. And then you've also got the jelly cytoplasm as well. Okay, so those are our structures of the animal and bacteria, and we're looking for differences. So, first one, both have a cell membrane. Well, yes, they both do, so that one is correct. Both have a cell wall. Well, our bacterial cell does, but our animal cell doesn't, so it's not that. Both have a nucleus. Well, bacteria don't have a nucleus, so it's not that. Both have cytoplasm, yep. And then both have plasmids. So the free DNA can also be called little loops of free DNA can be called a plasmid. Only bacteria have that. So it would be both have a cell membrane and both have cytoplasm. Salmonella is food poisoning is caused by bacteria in food. Give one symptom of salmonella food poisoning, but you can't say vomiting or diarrhea. Okay, so this is quite a common question. I won't give you the obvious answers, so you've got to think, what else happens to you if you have food poisoning? Well, the answers would be a fever or stomach cramps. Okay, you are allowed to say with fever a high temperature. So either of them would get you the 1 mark. Question 5.3. What is the name of the first antibiotic that was developed? This is just a little fact you need to know. It was penicillin. During World War II, that helped prevent loads of infection. A child with a severe bacterial infection was given a course of antibiotics. Figure 7 shows how the concentration of live bacteria in the child's body changed when taking the course of antibiotics. So you've got the time in days, you've got the course of antibiotics started at 0, and you've got the concentration of the of the live bacteria in the body, okay, on the y-axis. The concentration of live bacteria in the body continued to increase after starting the course of antibiotics. Suggest one reason why. So you have to, again, think about a logical reason. When you first start taking a course of antibiotics, why does the number actually start still going up? Well, it might just be that there's a time delay. Okay, but it takes a while for the antibiotics to actually then kill the bacteria to start seeing a difference in their total number. Okay, so there's a time delay for it to kill bacteria. It just might not be instant. Okay. After 3 days of taking the antibiotic, the child felt better, but there was still bacteria in the child's body. Why did the child feel better? The bacteria had become immune to the antibiotic, the child had become resistant to the bacteria, or there were fewer toxins in the body than at day 0. So if the child is feeling better, if the bacteria had become immune to the antibiotic, would that help the child feel better? No. That would help the bacteria be resistant to the drug. So that wouldn't make a child feel better. If anything, if they had become immune to the antibiotic, the child would feel worse. If the child had become resistant to the bacteria, well, straight away, not necessarily just because the child might now have antibodies to the bacteria, it might not make them feel better that quickly. But there are fewer toxins in the body than at day 0. This is more likely to be the correct answer. It's because, remember, if you've got fewer bacteria, the bacteria produce little chemicals called toxins, which then travel round our bloodstream and then can damage our cells and tissues. So if you've got fewer bacteria, you've therefore got fewer toxins. Suggest why doctors do not give antibiotics to patients with minor infections. Well, it's to stop antibiotics becoming less effective, or to prevent antibiotic-resistant bacteria developing. Okay. Yeah, that'd be your key answer on that one. That's also why we don't prescribe them for viral infections, because they only work on bacteria. So figure 8 shows blood viewed through a microscope. Okay, so you've got A, B, C and D. So they might give you different pictures of the components of blood. You've got to know what each one looks like. A is a red blood cell. B, the bigger ones, are white blood cells. C, the liquid that they're floating in is called plasma. And then D, the cell fragments, so not full cells, just little fragments of cells, are called platelets. A vaccine will stimulate the production of antibodies. Which part of the blood produces antibodies? So this is when you've got to know their functions. Red blood cells carry oxygen around the body. White blood cells are your immune system. Plasma is basically the liquid that carries everything else like CO2 and glucose and hormones. And platelets clot the blood, okay, helping us form scabs. So antibodies are part of our immune system, so it would be B, for white blood cells. Which part of the blood in figure 8 starts the clotting process? Well, we know that D, the platelets, they help clot and make our scabs, so it would be D. Question number 6. This question is about cell division. Write the biological structures from the box in the correct order of size. Well, hopefully, if you don't know this, you can at least identify the biggest one, and therefore it would be the cell. So the overall thing, because each of these things are contained in the cell. Now you should also know there's the nucleus is one of the main components in the cell. So just by using a little bit of common knowledge on cells, you can get the first two. Now, if you're not sure on the next two, because DNA is often taught a lot more in biology paper 2, but you should know that a gene is a short section of a chromosome. Okay, so if you're not even sure about that, you can take a 50-50 guess on that. But remember, if this is one of my pairs of chromosomes, a short section of a chromosome is a gene. But there's a lot more knowledge of DNA and cell, DNA structure in biology paper 2. Figure 9 shows how a fertilised egg can produce specialised cells. So you have a fertilised egg cell, and then it's gone into an embryo, and then process A is happening, which is turning it into red blood cells, muscle cells, and nerve cells. You've got to name process A. Now, a lot of people might put, they're becoming specialised, so therefore process is specialisation. That is not what the exam mark scheme is after. The embryo, remember, are stem cells. They are embryonic stem cells. And embryonic stem cells are undifferentiated cells. They have the ability to turn into specialised cells, such as red blood cells, muscle cells, or nerve cells. But this process is called differentiation. So you have to call the process of a cell becoming specialised, one word you've just got to know and be able to recall, differentiation. Question 6.3. How many divisions are needed to form a 16-cell embryo from the original fertilised egg? Okay, so this is when our knowledge of cell division comes in. We've got one cell, that then splits and becomes 2, and then 2 will become 4, and then 4 become 8. So we need to know how many divisions are needed. So 1 to 2 is 1 division, then 4, then 8, then 16. So you have 1 division, 2 division, 3 divisions, 4 divisions. Okay? Remember, each cell, when it divides, splits and makes 2 new cells in that process. In humans, a fertilised egg cell contains 23 pairs of chromosomes. So remember, it's already been fertilised, so the sperm's gone in, got 23 pairs. How many chromosomes will there be in each of the embryo cells? Well, it's already told you that you've got 23 pairs, you get 23 from the sperm and 23 from the egg. So therefore, it gives you a total of 46 chromosomes in those cells. Question 6.5. Figure 10 represents a cell cycle for a human embryonic cell. So you've got the cell cycle and you've got the 3 different stages here. Stage 1, stage 2 is mitosis, and then stage 3. Describe one change in the cell that occurs during each stage of the cell cycle. Okay, so it's basically saying what is happening in each stage of the cell cycle. So there are various things you can say. In cell 1, remember the cell has to prepare to divide. So in these ones, you could either say the cell has to grow or cell growth. It increases the number of mitochondria and ribosomes. It also has to duplicate or copy the DNA. Because remember, one cell's becoming 2, so therefore 2 copies of the DNA are needed. Then, stage 2, mitosis. When everything is copied, this is when the DNA actually divide. Okay, so it's the chromosomes are pulled apart to each side of the cell. That actually is the mitosis part of the cell cycle. So the DNA, after it's been copied, dividing. And then finally, in stage 3, when you've got everything you need, you've got the more mitochondria and ribosomes, the final stage, stage 3, is when your cell membrane and cytoplasm split, okay, forming 2 identical cells. So you could say any of these things you're allowed to say any of these things for your stage 1, any of these, chromosomes splitting for stage 2, and the cell membrane and cytoplasm splitting for stage 3. You could also say in stage 3 it makes 2 identical daughter cells. Right, last little bit of this paper then is the final 6 marker. Cell division is important in the growth of multicellular organisms. Figure 11 shows the mean height of boys and girls from birth to 18 years. Okay, so you've got the age in years and the mean height in centimetres for boys and girls. The question says, compare the growth of boys with the growth of girls using data from figure 11 in your answer. So you've got to say, key word here, compare boys and girls, and we've got to quote data. So this is a classic question where you actually haven't got to know anything. You've got to use this graph and extrapolate as much data as possible between boys and girls. So what I'm going to do, I'm just going to bullet point some facts around the outside of this graph of some key things that you can say. And what you want to be doing is making as many comparisons as you can. My favourite word when you're doing any comparisons are whereas. Remember, this is not English. You don't have to use all different fancy connective words. You can use the same comparison word over and over, as long as the points you are making make sense. So I would start off, treat it like a story. Start from the beginning of the graph and work your way across. Okay, so my first point that I'd be making is treating it like a story, going from 0 years and reading along, is all the way between 0 and then going down, pretty much halfway down, about 11 years, the boys are taller than the girls. So I would say the boys are taller than girls between 0 and 11 years. I could then potentially try and work out exactly how much taller they are. So looking at the gap between them, I can see that on average it's one little square going up, So therefore, if I work out how much each little square is worth, it would be 20 divided by 5, which is about 4 centimetres. So roughly 5 centimetres taller than the girls. You can then see they then cross at this point here. So I'd make that another key point. At 11 years of age, boys and girls are the same height. And I can even say exactly what it is. It's 140 centimetres. So you make that using data from the question. I'm referencing this key point here. Boys and girls are the same height at 11 years at 140 centimetres. Okay, then I would keep my story going, and I would say between 11 years and 14 years, you can see the rate of girls' growth is much faster than the rate of the boys. So between 11 and 14 years, girls' growth increases faster than boys. This thing happens up until 14 years, where they are the same height again. Until 14 years, when boys and girls, reading off data again, are both approximately 160 centimetres. After 14 years of age, we can then see it's the boys' rate of growth that increases, whereas the girls then plateaus. The boys increase from 14 to 16, and they finish just below 180 centimetres to finish at 179 centimetres approximately, whereas girls' rate slows and plateaus at just above 160, between 160, that would be between 160 and 164, so about 162 centimetres. Okay, so what we've kind of done, taking the points from the beginning, we've said what is happening at the beginning of this graph. We've clearly referenced the point where they cross. You then talked about the next part of the graph, again reference the points where they cross, and then the final parts of the graph where boys grow a bit faster than girls. Now, if you just fill your answer with loads of comparisons and points and fill it with loads of data, basically they're looking to see, can you interpret the graph and describe what the graph is actually showing. Okay, so you take all of those points and just write it in loads and loads of kind of short bullet point statements using whereas as a good connective word. I'm sure you do very well on that question. And that brings us then, there's one more question. Give one way that cell division by mitosis is important in fully grown animals. Well, we know that mitosis is needed for growth and repair. Okay, that is the function of cell division. We need it for growth and repair. Well, we can't say growth if they are fully grown, so therefore our final answer must be repair of damaged cells. So if our cells get damaged, they can be replaced through mitosis. And that then brings us to the end of the paper. So what I would suggest doing is, after you've kind of self-marked this paper or gone through it, identify any different areas you weren't familiar with, and then use the Cognito links at the bottom of this paper to improve your knowledge, and then try a different past paper that might have some similar questions on it. Okay, hope it's been useful. Thank you very much.